Hello, everybody. Today we are going to continue with our week of connections. Today we are going to make some connections between what we learned about chemistry last week and cell membrane structure, and start talking just a little bit about how things move in and out of cells. Yesterday, you looked a little bit at the molecules that make up the cell membrane. So see what you remember and see what you could figure out about that. The cell membrane, you might remember, is made up of a phospholipid bilayer. The primary part of it is two layers of those phospholipids. And you spent quite a bit of time yesterday looking at the chemistry of those molecules. When we describe the structure of a cell membrane, we often refer to it as a fluid mosaic model. So there are kind of two parts to the cell membrane. There's the part that moves around and is fluid, like a liquid, and there's the mosaic part, kind of this patchy mosaic of different molecules that make it up. So we're going to look at each of those aspects separately. When we talk about the fluid part, we're referring specifically to that phospholipid bilayer. Phospholipid molecules are what we call amphipathic. That's a big fancy word that just means they have some portions that are hydrophobic. That's the tails that hide in the middle of the membrane. And some parts that are hydrophilic. That's those heads that stay towards the outer part of the membrane where there's water. This is a fluid membrane because these molecules can slide past each other. And there are a few different aspects of the chemistry that contribute to this fluidity, this ability for the membrane to kind of flow around the cell. One of them is the temperature. And this should kind of make sense. If you think of any kind of lipid that you know, anything like butter or wax or oil, the temperature affects how fluid it is, right? Lower temperatures tend to make membranes less fluid, just like if you chill butter, it gets more solid, or if you cool bacon grease, it congeals and becomes more solid. And if you heat things up, higher temperatures are then going to make them more fluid. The molecules start moving faster, and so they slide away from each other a little bit. Just like if you heat up butter or oil or anything else, it melts and becomes more liquidy. The same is true of a cell membrane. The chemistry of the molecules that makes up the membrane also affects how fluid it is. The more unsaturated fatty acid tails there are, remember, unsaturated fatty acids have a double bond that causes that tail to kind of kink and bend. And when those tails are bent, they can't pack as closely together. And so they have more space and more room to move around, which makes for a more fluid membrane. The other part of the membrane is cholesterol. You learned yesterday that cholesterol is another lipid that's found in cell membranes. But if you look at the structure of cholesterol, cholesterol is a type of steroid, and it takes up more space. Remember, steroids have that 4-carbon ring structure. And because it takes up more space at lower temperatures, we'll start with that side. At lower temperatures, that bulkier molecule can't pack as closely as all those nice, neat phospholipid tails. It takes up more space. It keeps the molecules farther apart, so it doesn't let the membrane totally solidify. But at higher temperatures, cholesterol actually forms bonds. They're weak bonds, but it forms bonds to a bunch of the phospholipids around it. So that helps hold the molecules together as the temperature goes up. So cholesterol kind of helps to moderate the fluidity of the membrane and keep it about where cells need it to be so that it doesn't get too fluid when it gets warm, but it also doesn't get too solid when it gets cold. Membranes have to be fluid to work. Generally about the consistency of like a salad oil, like salad dressing. So you really can imagine a cell membrane like a bubble of oil that surrounds the cell and is kind of flowing and moving around the outside of the cell. The mosaic part of that fluid mosaic model refers to the proteins that are found embedded within or on the surface of the cell membrane. This picture just shows you proteins that go all the way through the membrane. There are also some that just sit on one side or the other of the membrane. Many of these proteins are also amphipathic. They also have hydrophobic and hydrophilic regions. Some of them can move within the membrane, but they tend to move more slowly than the lipids do. And others are anchored in place, either by being attached to the cytoskeleton inside of the cell or something called the extracellular matrix that you'll learn more about later on the outside of the cell. The proteins that are found on the surface of a cell determine the function of that membrane. And sometimes you'll find different proteins kind of in patches in different areas of a cell membrane to help the cell membrane perform different jobs. The proteins that go all the way through the lipid bilayer are called integral proteins. And so these proteins have hydrophobic regions. Remember, proteins are made of amino acids, which have all those different R groups with different chemistry. The amino acids in the central part of these proteins would have R groups that are hydrophobic, that will sit nicely within that hydrophobic region of the bilayer. The ones towards the outside would have R groups that are more hydrophilic. Peripheral proteins are the ones that are bound to the surface, either on the outside of the cell or the inside. These proteins have a huge variety of functions. You see a few of them here to anchor the cell in place. They can be enzymes that help catalyze reactions. They can help with signaling functions. They can help transport things. And we'll talk more about a lot of those proteins as we continue through biology. Cell membranes, in addition to the lipids and proteins, also contain carbohydrates. So they really have 3 of the 4 main groups of biomolecules. The carbohydrates mainly assist in communication between cells, helping cells to recognize each other. This is important for organizing your tissues within your body. It helps your heart cells recognize other heart cells and lung cells recognize other lung cells. It's also important in your immune system, and we're actually going to come back to this a little bit later this week as we start talking just a little bit about viruses in your immune system. We'll come back to the role of some of these carbohydrates. Usually the carbohydrates are short branched chains. You can see them in blue on here. If they're attached to the lipids in the cell membrane, then that combination of molecules is called a glycolipid. The glyco part refers to the carb, and the lipid, obviously, is that phospholipid. If they're attached to a protein, we call that a glycoprotein, that combination. The carbohydrates on the outer side of the membrane can vary from one species to another. They can also vary between individuals of the same species. When you talk about ABO blood types, you're talking about these carbohydrate chains on the surface of blood cells of humans. And they can also vary between cell types within your body. So they're used for a lot of identification and communication at a lot of different levels. So we have been looking at the cell membrane in this potato osmosis lab that your classmates started yesterday. The way this lab is set up, this picture just kind of shows you what this setup looks like. There are 6 different solutions that were provided, and they were just labeled A through F. There's a number 1 next to these just so that I remembered which set these were. Each of these is just a mixture of sugar water of different concentrations. They look yellow because I added yellow food coloring, so you can't tell which is which. And they range from no sugar at all to a 1 molar sugar solution. And part of your task in this lab is going to be to figure out which of these solutions is which concentration. They do not necessarily go in order. The way that we're going to figure that out, yesterday, your classmates took the mass of 3 pieces of potato and put them into each of these solutions. And then today they're going to take the mass again after they've been sitting in there for about 24 hours. That will allow us to determine which of these potatoes gained mass and which of them lost mass due to the movement of particles across the membranes of the cells of those potatoes. And that'll help us figure out which concentrations are higher and which concentrations are lower. This is a lab that we will come back to when we learn more about osmosis and water potential in another week or two. So we're going to do kind of a preliminary analysis. You're going to take your best guess at an explanation of what's happening here this week. And then we will return to this data and look at this again in a little more depth in another week or two. So this is the procedure that the students in class followed today. As they were gathering their data, you'll notice at the bottom there will be a class data table that will be linked with the potato osmosis lab that you will need to access and look at when you are completing the post lab. But also, in order to complete this post lab, you're going to have to think a little bit about movement across cell membranes, particularly diffusion and osmosis. So let's review just a little bit of basics about these. Both diffusion and osmosis are types of passive transport, which means they happen without any energy being expended by the cell. Diffusion happens because individual molecules move randomly, but if there's a large concentration of them in one area as a group, they may have a direction and move down their concentration gradient. Things will always move from where there is a higher concentration of that molecule to where there's a lower concentration. A concentration gradient is actually a form of potential energy. Think of it just like the slope of a hill. Something at the top of a hill has gravitational potential energy because it has the potential to go down that hill. A concentration gradient represents chemical potential energy. Where there's a high concentration of molecules, they have the potential to diffuse and move kind of downhill to where there's a lower concentration. This is an idea that's going to come back several times as we talk about some important chemical processes in biology. So here are some examples of some types of molecules that may need to move in or out of cells. Take a minute first and see if you can complete those boxes on the bottom to talk about whether these molecules are polar or not, and whether you would classify them as small or large. So the first column, the type 1, are ions. So those are actually charged particles. We don't just call them polar, they're actually charged. Glucose is an example of a polar molecule. It has lots of hydroxide groups there, lots of oxygens in there that cause it to be polar. The type 3 molecules include water, which you hopefully remembered is a polar molecule as well. The bottom row here talks about the size. Ions are very small. It's just one atom or sometimes a couple atoms that are positively or negatively charged. Glucose, on the other hand, is quite a large molecule. Water, by contrast, is pretty small, and so is oxygen and carbon dioxide. They're just a couple of atoms bonded together. So as we think about these in terms of cell transport, which of these 4 types of molecules would most easily diffuse through the cell membrane on its own? Think about the chemistry of the cell membrane as you answer that. So hopefully, and I'll move my picture out of the way a little bit, you realized that molecules that are small and nonpolar are the ones that are most easily going to be able to diffuse through the lipid bilayer. In order to get through that hydrophobic interior, these molecules need to be nonpolar to slip through those hydrophobic tails without any problem. So things like oxygen and carbon dioxide can easily diffuse in and out of cells from high to low concentration. Other types of molecules like water and urea, these are also small, but they're polar. And a polar molecule is not easily going to go through the hydrophobic interior of the cell membrane. These types of molecules often diffuse through a protein channel. This is called facilitated diffusion. So here's an example. This is a protein called an aquaporin that specifically allows water to diffuse through. And this protein has R groups that kind of line the interior here that make this channel polar so that water can more easily travel through, and it doesn't have to go through that hydrophobic part of the cell membrane. It can go through this nice hydrophilic part so that water can diffuse easily in and out of cells. Larger molecules like glucose usually can't cross the membrane on their own. They usually need assistance from a protein, and they usually require energy. So now we're talking about active transport. Sugar will not cross the membrane on its own. And that last column, the type 1 ions, either need to have help from a protein or they need to be pumped across the membrane by active transport, depending on the situation. We will come back and talk about all of this more later as we get into distance learning and into Unit 4. But this is some important stuff to think about because, like I said, the potatoes have been sitting in sugar water. So particularly here, think about what we just said about water and sugar and their ability to move across the membrane. And that will help you to try and figure out what's going on in that potato osmosis lab. So for the next few days, you have 2 things here to be working on. The first part of this potato osmosis lab is due by Friday. Think of this kind of as a rough draft. I want you to think about it. You may need to look up a couple vocab words to help you figure it out, but try not to just copy something you find online. Really try and think about it and understand and explain what's happening as best you can. You are going to have a chance to come back and revisit this and change some of your answers when you learn a little bit more about osmosis in a couple weeks. But this first draft, this first post-lab part is due by Friday. You also have a Mastering Biology homework that is due tonight, so you can go and work on that now if you are not finished with it. Please email me if you have any questions, and I hope you have a great day.